Hello everyone, I am Prashant and now I am going to discuss about the topic which is genome organization, gene expression and its regulation. So in human body, the genetic information is known as is stored in the DNA. So the genome is defined as the complete set of genes or the genetic information in an organism. So and the segment of the DNA which is expressed to yield a functional product. That functional product may either be an RNA or a protein. So that segment of the DNA is known as a gene. And genome organization is the study of the sequential arrangement of genetic information in the body. So that first we will see how the DNA is formed and how it is packed. The DNA are the formed by the polymers which is synthesized from the building blocks called nucleotides. This nucleotide consists of a nucleobase, a sugar, and one or more phosphate groups. That base may either be purine or pyrimidine. The example for purine base is adenine and guanine. For pyrimidine base, the example is cytosine and thymine. The RNA and DNA differs in the structure of the sugar. That is, in RNA it consists of a ribose sugar, whereas in DNA it consists of deoxyribose. This base and this nucleobase and the sugar is together called as nucleoside. This nucleoside may consist of one or more phosphate groups, and this nucleotide is joined by phosphodiesterase linkage between 3' OH of one nucleotide to the 5' OH of the another nucleotide. Each nucleotide is added with a 3 to 5' linkage in the same way. This sugar and phosphodiesterase linkage comprise a monotonously repeating sugar phosphate backbone, and the base vary from one nucleotide to another. As you can see that the A represents A adenine, and T represents thymine, G represents guanine, and C cytosine. And this nucleobase varies from one nucleotide to another nucleotide. And DNA is double-stranded. That is, the bases on opposing strand form hydrogen bonds that cause them to pair with one another. The cytosine form three hydrogen bond with guanine, and thymine forms two hydrogen bond with adenine. And this basing is known as the base pairs, and these base pairs are paired in opposite direction, and hence they are known as anti-parallel direction. And the length of the DNA is measured in the form of base pairs. And this DNA binds to form a double helical structure, and this double helical may organize in many forms such as bDNA, zDNA, and aDNA. The most common organization is bDNA, and the length of the one turn of this bDNA is around 10.5 base pairs, and the distance between two base pairs is known as 3.4 angstrom. And this bDNA consists of two grooves, that is major groove and a minor groove. So now we will see how the DNA is packed. The length of the DNA in one cell is around 2.2 meters, but the diameter of our nucleus is 1 micrometer, that is 10 power minus 6 meters. So the packing of this DNA in that small nucleus is very complex thing, but this DNA is packed with the help of a protein known as histones. These histones are the proteins around which the DNA binds to create a structural unit known as nucleosomes. These histones are of 5 types. This H1A, H1, H2A, H2B, H3 and H4. Among these, H1 is known as the linker histones and another 4 are known as the core histones. These core histones consist of a positive charge on lysine and arginine residues, and this positive charge counterbalances with the negative charge of the phosphate group of the DNA. This core histones forms an octamer unit, and the DNA wraps around this octamer unit around 2 or 1.6 times, and that is for around 146 base pairs the DNA wraps around these histones, and this histone molecules is connected with another histone molecules with the help of the linker histones, that is H1, and there is a space between one histone to another histone known as linker DNA, which is around 80 base pairs, and the diameter of this histone is around 10 nanometer. This histone forms a bead on a string form of chromatin, and this wraps around one another to form a solenoid structure, and it is around 13 nanometer of diameter, and this solenoid structure form a thin thread-like structure called chromatin. This chromatin is highly condensed to form a chromosome. Hence, chromosome is the chromatin fibers which is condensed and folded into much larger and compact structure. In human, there are 23 pairs of chromosomes, out of which 22 are autosomes and one pair of sex chromosomes. That is double X in female and X and Y in male. And these chromosomes consist of a heterochromatin and euchromatin. In heterochromatin, the histones are densely packed and they are not actively available for the transcription. Whereas in euchromatin, the histones are less densely packed and they are transcriptionally active. So now we see what are the elements present in the human genome. So as I said that the DNA in human is present both in nucleus and in mitochondria. The nuclear genome or nuclear DNA consists of a few copy sequence or single copy sequence, and it consists of repetitive elements. In mitochondrial genome, the DNA consists of 2 ribosomal RNA molecules, 22 tRNA genes, and 13 polypeptide encoding genes. The coding elements in nuclear genome, the around 98.8% is non-coding and only 1.2% of DNA is coding. That is, this 1.2% is a functional protein molecule. And the coding elements in the nuclear genome is called as exons, or it is also called as expressed sequence. The non-coding elements is called pseudogenes, and pseudogenes, these are the genes of multiple copies, and these are mostly non-functional, and it also consists of introns. These introns are present between the exons, and this also called as intervening sequence, and it is initially copied into RNA, and these are cut off from the final RNA transcript. And it also consists of a UTR, or also called as untranslated region, and this is also called as spacer sequence. These are the non-coding sequence between genes, that is, it connects one gene with the another gene. It consists of also repetitive elements. The repetitive elements are of two types, that is short randomly repeated elements and interspersed repeated elements. These randomly repeated elements are the multiple copies arranged one next to another. These are also called as satellite DNA because the base composition of these randomly repeated elements appear in a satellite band. And here is the example of the randomly repeated DNA sequence, that is this GTTAC, this 5 codon or 5 base are repeated next to one another. These randomly repeated are of two types, that is mini satellite DNA and microsatellite DNA. Mini satellite DNA are of around 100 to 10,000 base pairs in total length, and this microsatellite DNA are 1 to 10 base pairs in length. This mini-satellite DNA are also called as variable number tandem repeats, or VNTRs in short, and microsatellite DNA called as short tandem repeats, or STRs. The next type of repeated elements are interspersed repeated elements. These are the repeated elements which are scattered throughout the genome. It is also called as junk DNA because the function of these repeated elements are not yet discovered. It is also known as transposons because they have the ability to move from one sequence to another sequence, and wherever they move, they leave the copies of themselves wherever they stop. These interspersed repeated elements are again of two types, that is line and sign. Line is known as long interspersed nuclear elements, which is around 6,000 to 8,000 base pairs in length, and these are mostly expressed to read a enzyme. And the next another type of repeated elements are SINE, also called as short interspersed nuclear elements, and these are of 500 base pairs in length. The example for SINE is allele sequence, which is less than 300 base pairs. And the DNA or genetic content is also present in the mitochondria. In mitochondria, the DNA or divide of it does not contain any histones, and it is mostly circular DNA. It consists of around 16,569 base pairs, and it measures around about 5 mm in length with 37 genes. The RNA and polypeptides encoded by this DNA are just a small fraction, and it consists of around it consists of around 13 of the polypeptide subunits of the electron transport system, And it also consists of subunits of NADH dehydrogenase, cytochrome B, cytochrome C oxidase, and ATP synthase. Genetic basis of disease. Most of the genetic disease is thought to be due to rare single gene disorders such as cancer, hemophilia, or phenylketonuria. Even though they are thought to be rare, nearly 1 in 17 persons are affected by them. According to geneticists, mutation is defined as any editable change to the DNA sequence. But in past, we thought that mutation means it is a disease state due to the change in DNA sequence. In modern practice, this mutation is commonly referred to as variants. This variants may be of two types, that is benign or pathogenic. Benign means it is not associated with any disease, and pathogenic means it is associated with the disease. So the main common genetic disease is due to single nucleotide polymorphism. It is the frequent change in our genome that is affected by only one base pairs, and it is estimated that at least 11 million SNPs are identified till now. And the most common example for SNPs are sickle cell anemia, cystic fibrosis, and thalassemia. And the next one is structural variants, and it is defined as the variants affecting segments of DNA greater than 1 kilo base pairs. And the example for structural variants are leukemia, which is due to translocation, and hemophilia A, which is due to inversions of the DNA segments, and next one is Cray-Duchat syndrome, which is due to large deletions of the DNA sequence. Complex diseases are those that are caused by a combination of multiple genetic and environmental factors. The examples of that complex disease are cardiovascular disease, type 2 diabetes, autoimmune disease, cancer, and Alzheimer's disease. So till now we have studied about the genome organization. Now let's move on to the next topic, that is gene expression. Gene expression, it is the process in which the functional product is obtained from the template DNA strand. The functional product may be from a protein, and this protein is synthesized by the steps such as the DNA to RNA transcription takes place, and from RNA to protein translation takes place. This protein is the final product of the gene expression. In few incidences, RNA will be the final product of gene expression, and it functions as such within the cell. So now let's study what is transcription and translation. This transcription takes place mainly in the nucleus, whereas translation takes place in the cytoplasm. Transcription, it is the first step in gene expression. It involves the synthesis of RNA from template DNA strand. The transcription takes place in 4 stages, that is RNA polymerase binding, that is the RNA polymerase should come and bind to the DNA, and from there it should initiate the transcription process, and next stage is elongation and the termination, that is it should end the transcription process. First one is RNA polymerase binding. There are majorly 3 types of RNA polymerase, that is 1, 2, and 3, and each synthesizes 1 or more classes of RNA, and each polymerase has 1 or more promoter regions in the DNA. Now, first type of RNA is RNA polymerase 1, and it is majorly found in nucleolus, and it synthesizes 28S RNA, 18S, and 5.8S ribosomal RNA, and it has 2 promoter elements, that is core promoter and upstream control element. And RNA polymerase 3, it is a nucleoplasmic enzyme. It synthesizes tRNA and 5S ribosomal RNA. The promoter for the synthesis of tRNA is box A and box B, and the promoter region for synthesis of 5S ribosomal RNA is box A and box C. And another type of RNA polymerase is RNA polymerase 2, and it is found in nucleoplasm, and the synthesis, it is the precursor for the synthesis of mRNA, which is the class of RNA molecules that encode proteins. This RNA polymerase 2 has 4 types of DNA sequences, which is mainly a promoter region, that is a short initiator sequence, and Tata box and transcription factor 2B recognition element, which is also designated as BRE, and another type of promoter region is downstream promoter element. So this is the next is general transcription factors. These are the protein which are required for a RNA polymerase molecule to come and binds to its promoter region in the DNA, and thereby it synthesizes the RNA. So each eukaryotes have many transcription factors, and the name include TF designated as transcription factor, and the Roman numericals such as 2, which tells us that it is a transcription factor for RNA polymerase 2, and it is been given a alphabetical letters to identify each transcription factors. The general transcription factors bind to an DNA in a specific order, and it mainly starts with TF2D. Other transcription factors primarily interact with each other before binding to the DNA molecule. A large complex of proteins, including RNA polymerase 2 and the promoter region, it is known as pre-initiation complex. Before initiation, RNA polymerase 2 must be released. This can be done by TF2H. This TF2H mainly acts as helicase and kinase activity. This helicase means it unwinds the DNA strands to form a transcription bubble, and kinase means it phosphorylates the RNA polymerase. That is, in RNA polymerase, it consists of a C-terminal domain which acts as a tail. Upon phosphorylation, this tail gets activated and the RNA polymerase gets initiated to start the RNA synthesis. The first step in and the next step in RNA synthesis is initiation. The RNA polymerase opens up a short region of 14 bases, and that region is known as transcription bubble. Free ribonucleoside triphosphate enters through the funneled region of the RNA polymerase and forms complementary base pairs with bases on template strand. First base is usually purines, and these nucleotides are successively added one after the other, and this addition is basically energy favorable, and there is a release of pyrophosphate upon each addition of nucleoside triphosphate. After 9 to 10 or 9 to 11 nucleotides, the enzyme breaks free from the promoter and forms a elongating complex. So in elongation, transcription proceeds in a processive manner, and nucleotide is added one after the another, and it forms around hundreds to thousands of bases. The transcription bubble of 10 to 12 unpaired bases is maintained, and the termination. After transcribing complete set of genes, enzyme must stop transcription at proper position and release completed transcript unit. By assortment of signals, this termination can be done. The cleavage site is around 10 to 35 nucleotides downstream from the special AAU units or AAA sequence in the growing RNA chain. So the next is post-transcriptional changes. The RNA which is produced by the transcription is known as primary transcript, and it is not functional, and it is processed by chemical modification to generate final RNA product which serves as a precursor for translation. And the first one is mRNA processing. The mRNA which is synthesized by transcription is known as pre-mRNA, and it should undergo a few changes to get converted into mature mRNA. And it is the mRNA processing is of two types, that is addition of 5 caps and 3 prime tails, and removal of introns. At 5 prime end of the mRNA, the guanosine nucleotide which is methylated at 7 position is added, and at the 3 prime end, adenine nucleotides poly A tail is added, and then after this there is a removal of introns. These introns are removed by the enzyme spliceosome, and it is Then the remaining exon part is attached to yield a functional mRNA unit, and then this mRNA is transported to the nucleus to the cytoplasm. And the next one is ribosomal RNA processing. The DNA sequences that encode these ribosomal RNAs are separated within the transcription unit by DNA called transcribed spaces. After the RNA is synthesized, the resulting pre-rRNA molecule is processed by a series of cleavage reactions that remove the transcribed spaces and release the mature RNAs to the cytoplasm. These ribosomes are built from two dissociable units called large and small subunits. That is, the 60S unit and 40S units are made together to form a 80S ribosome subunit. Next is tRNA processing. Cells synthesize several dozens kind of tRNA molecules, each designated to bring a particular amino acid to one or more codons in the mRNA. A mature tRNA molecule contains only 70 to 90 nucleotides, some of which are chemically modified. Base pairing between complementary sequence located in a different region causes each tRNA molecule to fold into a secondary structure containing several hairpin loops. It also contains 4 base paired regions and a variable loop. The leader sequence is removed, and then the replacement of nucleotides with the CCA group is done, and then the chemical modification of bases is done in the variable region, and the excision of the intron here from the purple region of the loop. So after transcription, the mature RNA molecules are transported to the cytoplasm to undergo translation. For translation, majorly 5 components are required, such as ribosomes that carry out the process of polypeptide synthesis, and tRNA, the molecule that assign the amino acids in the correct order along the mRNA template, and amino acid tRNA synthase that attach amino acids to their appropriate tRNA molecules, and mRNA molecules that encode the amino acid sequence information for the polypeptide to being synthesized, and it requires protein factors that facilitate several steps in the translation process. So the translation process is divided into 3 stages, that is initiation, elongation and termination. So for initiation, it is a complex thing in eukaryotes, and it requires 12 protein factors which are designated as eukaryotic initiation factors, that is EIFS. Pre-initiation complex is formed by 40S subunit which is binded to initiated tRNA molecule and EIF1. mRNA is brought to the ribosome by EIF4 group of factors. The 5' cap of mRNA recognized by EIF4E and forms complex with 4A and 4G, and this 4G molecules also forms complex with the poly A binding protein of mRNA molecules. The 40S subunit scans mRNA to identify AUG, which is called as initiation codon, and when AUG codon is reached, the EIF5 triggers hydrolysis of GTP bound to EIF2. The initiation factors are released, and the 60S and 40S subunit forms a complex to form a 80S ribosome subunit. And after initiation, elongation takes place. The ribosome has 3 sites for tRNA binding, that is peptidyl site, amino acid site and exit site. The first step in elongation is binding of the next amino acid tRNA to A site by pairing with the second codon of the mRNA. Based on the codon, the correct amino acid tRNA binds to the tRNA at P site, and translocation occurs by moving 3 nucleotides along mRNA. Elongation continues until a stop codon, that is UAA, UAG or UGA is reached. Because there is no tRNA molecules to code for this codon, and after the UAG or stop codon is reached, the elongation release factor binds to the codon at the A site and stimulate hydrolysis of bond between tRNA and polypeptide chain at P site, and everything such as 60 a ribosome unit dissociates into 60S and 40S, and the mRNA molecules dissociates and tRNA molecules also dissociate, and a polypeptide chain which is synthesized is also dissociated, and the translation process finally comes to an end. So the next topic is protein folding and processing. The translation completes the flow of genetic information within the cell. The sequence of nucleotides in the DNA has now been converted to the sequence of amino acid into a polypeptide chain. The synthesis of a polypeptide is not equivalent to the synthesis of functional protein. To be useful, these polypeptides must fold into distinct 3D conformation, and in many cases, the polypeptide chains must assemble into a functional complex. In addition, many proteins undergo further modifications, including cleavage and covalent attachment of carbohydrates and lipids that are critical for the function and correct localization of proteins within the cell. So, we'll study about the chaperones. These are the proteins which facilitate the folding of other proteins, and it acts by binding to the amino terminal portion of the nascent peptide chain. And this completed protein is then released from the ribosome and is able to fold into its correct 3D conformation. If the protein is not correctly folded, it may cause dire consequences, because some misfolded proteins can aggregate and form insoluble fibers called amyloid fibers that accumulate in extracellular spaces and within cells. So these amyloid fibers can cause many neurodegenerative disorders such as Alzheimer's and Parkinson's disease. There are basically two types of chaperone proteins, that is HSP70 chaperones and chaperonins. These proteins bind to the short hydrophobic segments of unfolded polypeptides, maintaining the polypeptide chain in an unfolded configuration, and it also prevents aggregation. The unfolded polypeptide chain is then transferred from an HSP7 chaperone to a chaperonin, within which protein folding takes place, yielding a protein correctly folded into its functional 3D conformation. So the next type of protein changes is by enzyme, that is catalyzing by enzyme. There are basically two types of enzyme, that is peptidyl prolyl cis trans isomerase and protein disulfide isomerase. Here you can see that there are incorrect disulfide bonds between the proteins. By enzyme called protein disulfide isomerase, the sulfide group is cleaved and the correct disulfide bonds are formed. And the next is glycosylation. Many proteins are modified by addition of carbohydrates, and this process is known as glycosylation. The proteins to which carbohydrate chains have been added are called as glycoproteins. And the next one is attachment of lipids. The types of lipid additions are N-methylation, prenylation, and palmitylation, and these are the most common. And there may also a fourth type of modification, that is addition of glycolipids, which plays an important role in anchoring some cell surface proteins to the extracellular phase of the plasma membrane. And the another change of protein changes are protein cleavage. Cleavage of the polypeptide chain is an important step in the maturation of many proteins, and it is also known as proteolysis. For example, the mature insulin molecule consists of two polypeptide chain which is joined by disulfide bonds, and this basically known as pre-proinsulin, and the joining of the disulfide bonds is called as proinsulin, and the additional sequence is in this proinsulin is then removed by connecting a polypeptide, and thus it forms a final functional insulin molecules. Regulation is an important aspect of almost every process in nature. This is especially true of gene expression. So gene regulation is the process which either increase the process of gene expression, or either they may decrease the process of gene expression. And gene expression can be regulated at many stages, and we mostly focus on two stages, it is transcriptional regulation and translational regulation. So in transcriptional regulation, the interaction of general transcription factors and RNA polymerase with the core promoter often initiate transcription at a low rate. In addition to a core promoter, most protein coding genes have short DNA sequences located in the upstream region to which other transcription factors bind, thereby improving the efficiency of the core promoter. If these additional DNA elements are deleted experimentally, the frequency and accuracy of transcription are reduced. Such regulated DNA sequences are often called as proximal control elements located upstream of the core promoter within about 100 to 200 base pairs. Transcription factors that selectively bind these sequences are called regulatory transcription factors. So, then enhancers and silencers. Enhancer are the sequence of the DNA to which certain transcription factors should bind, and they stimulate the gene transcription. These are also known as transcriptional activators. These are located at the variable distance from the promoter, and silencers are the sequence that inhibits the transcription, and these are also called as transcriptional repressors. And insulators are the sequence which restrict the influence of enhancers and silencers. Coactivator. These are the sequences which are located at the very far region from the core promoter region. It occurs at very many stages. A group of activator proteins bind to the respective DNA control elements within the enhancer, forming a multi-protein complex called enantiosome. One or more of this activator protein causes the DNA to bind and creating a DNA loop that brings the enhancer close to the core promoter. The activator interact with coactivators such as chromatin remodeling proteins and histone acetyl transferase, which alter chromatin structure to make the DNA in the promoter region more accessible. Finally, the activators bind to mediator, which facilitates the correct position of RNA polymerase and the general transcription factors at the promoter site, and thus allow transcription to begin. So the next type of regulation is translational regulation. The translational regulating proteins. It is facilitated by binding of the repressor proteins to specific mRNA sequence, which blocks translation. The example for this type of mechanism in eukaryotes is regulation of synthesis of ferritin. Ferritin is a protein that stores iron within the cell. The translation of ferritin mRNA is regulated by supply of iron. If there are more ferritin is synthesized, if more ferritin is synthesized if iron is abundant, this regulation is mediated by protein which binds to a sequence that is iron response element in the 5' untranslated region of ferritin mRNA, blocking its translation. In the presence of iron, no repressor binds to the IRE and ferritin translation is able to proceed. Translation repressor binding to 3' untranslated sequence. The translation repressors can bind to the regulatory sequence in the 3' untranslated region and inhibit translation by binding to an initiating factor EIF4E bound to the 5' cap. This interferes with the translation by blocking the binding of EIF4E to EIF4G in a normal initiation complex. The translation is also globally regulated in response to amino acid starvation. The protein synthesis is regulated in response to external conditions and stimuli. It should be high when nutrients are plentiful to allow maximal cell growth and low in times of nutrient deprivation. Under normal conditions, the tRNAs are immediately amino acylated and once released from the round of elongation on the ribosome and are bound in the cell by elongation factor thermo-unstable unit once they are loaded with appropriate amino acid. The uncharged tRNA is an indicator of low levels of amino acid and trigger for global shutdown of protein synthesis in both eukaryotic and even prokaryotes, although their mechanism of response are different. The phosphorylation of an initiation factor regulates protein synthesis globally. The eukaryotic cells decrease their overall rate of protein synthesis in response to variety of stressful situations, including deprivation of growth factors or nutrients, or infection by the viruses, and sudden increases in temperature. Much of this decrease is caused by phosphorylation of translation initiation factor EIF2 by specific protein kinase that responds to the changing conditions. So, the gene misregulation in the disease. The gene expression in humans are controlled by number of transcription factors, cofactors, and regulator. Misregulation of this gene expression may lead to broad range of diseases such as cancer, autoimmunity, neurological disorders, developmental syndrome, diabetes, cardiovascular disease, and obesity, etc. So, this comes to the end of my topic, and these are the references which I referred for my studies, and thank you.